Celiacincria Symposium Romano-German pentru Gastroenterologie. Thank you very much for inviting me here. I learned this 40 years ago, the last time I was here as a very young student. Thank you. I come from one of the very oldest universities in Germany, as you can see here. And this is located, many may not know where it is. It's right here on the Baltic coast and a little distant away from Sibiu. As a disclaimer, I'm not going to talk about any drug products, but we do have a beach in Greifswald, which is right here, belongs to the hospital, and in the far distance on the horizon you see the island of Usedom. And there the most famous person from Sibiu is not Paul Pohr, but Hermann Obert. He worked for a while in Peenemünde. This is his product. He wanted to shoot people into space. And unfortunately, Hitler made him to develop the V2 bombs rather than rockets, but he later got his chance to do rocket. I will talk about pancreatitis, and I will give you a little background on pancreatitis first. I will allude to what we did and now do with pancreatic necrosis because it has essentially become an endoscopic therapy. And I will also talk about some of the translational science I do in my lab, which is very much concerned with inflammation and inflammatory cells, which we think is responsible for the severity of pancreatitis. So pancreatitis is an inflammation of the pancreas whose onset is not usually caused by infectious agents. So it starts by very different mechanisms, although later in the disease, bacterial inflammation is probably what kills the patients, and the defense against this bacterial inflammation is key to future therapies. The most frequent reason of hospital admission in the US is acute pancreatitis among all benign GI diseases. So the cost burden is enormous, and in the US you have around 240,000 hospital admissions for acute pancreatitis alone. And the interesting thing, we do not understand that 80% of patients with acute pancreatitis go home after a few days of pain treatment and fluids, and 20% develop necrosis, which is an indicator of very severe disease. And they develop complications, and they are the one we're concerned about because they have a lethality rate of 5 to 20% in the hospital. In Germany alone, this is 1,500 deaths from acute pancreatitis. If you consider it, it's twice the number of AIDS patients that die in German hospitals. If you still consider the research money that goes into AIDS research and to pancreatitis, then you have a little disparity. Part of my training I did in Ulm, and what I learned in Ulm, where they had a very, very potent surgical department, is from this study of Markus Büchler and Hans Beger, where necrosectomy was the rule. This is probably one of the most important papers of 30 years ago. They they investigated 114 patients with acute necrotizing pancreatitis and operated on all of them. They did a necrosectomy, and the seminal finding they had was that the post-operative mortality was 40% if they found bacteria in the necrotic material. It was only 8% with negative cultures, and the post-operative mortality, and this is key, was if you operated early in those patients and they had septicemia, with or without bacteria, they all died. So the conclusion from the study was infected necrosis is the one that kills the patient, particularly when it develops in the early disease phase. What was not the conclusion, and should have been, is don't operate on patients with sterile necrosis, and certainly don't operate on them early. That was then, of course, challenged over the years, and after Ross Carter from Glasgow had introduced a new technique of debridement of infected necrosis, which involves putting a catheter in from the flank, dilating the tract, and getting the necrosis out in a minimally invasive way, mortality suddenly dropped. So one of the first really randomized clinical trials to compare the then standard open surgery for infected necrosis versus minimal invasive surgery was done by this fantastic group, the Pancreatitis work group of the Netherlands. They received 5 million annually from oil revenue from the Dutch government, and they put up a fantastic group of 20 centers who really operate and put out the most seminal study. They compared a step-up approach, which means first drain, and if needed, do minimally invasive surgery, compared to open surgery. And what they found is that minimally invasive surgery is much better than open surgery, and what was even more surprising, 35% of patients with infected necrosis only needed drainage. And that was a real step forward, and it made us believe that minimal invasive necrosectomy is superior to open necrosectomy, and more important, often not even needed. The next step was, this young man, then young man, came along, Hans Seifert, now in Oldenburg. He had serendipitously discovered that he accidentally made a hole in the wall of the stomach in severe necrosis, and when he was there in the retroperitoneum, he took the necrosis out. This is what endoscopists do. There's an accidental hole, you go in and you take the stuff out. But then he put a systematic trial on top of that. He had three more cases, and that was the first step to endoscopic necrosectomy. He even states in his initial paper, the standard of management of infected pancreatic necrosis then is open surgical debridement. And he proposes on 3 patients that this may be done endoscopically, and pioneered this technique. The questions that remained open is, how safe is this procedure? And Hans himself said later with more experience, you can only do it under CO2 atmosphere, and he defined the complications. The question was how safe it is, what patients should be selected, how many often did you have to repeat it? Is it enough to just take the plug off and get the pus out, or do you have to have a very serious debridement until everything is clean, and how to maintain the fenestration? And that is the latest news from this January, just a month ago. Again, the Dutch pancreatitis group with Sandra van Bronschoot here as the PhD student who did this, they compared a composite endpoint of death and major complications in minimally invasive, surgical, from the retroperitoneal access, with endoscopic debridement in severe pancreatitis, and the primary endpoint was not different between the 2 groups. However, because the number was not high enough to show that between 20 and 28% there is a difference, the interesting thing is, again, half of the patient didn't even need a debridement once you put a drainage in and you get the pus out. However, the rate of fistula, the length of the hospital stay, and the cost was actually much higher in the minimally invasive surgical group. So there is an advantage for endoscopic necrosectomy in acute necrotizing pancreatitis. The lesson is you should do endoscopy first. It's cheaper, it's as safe, and it's probably as effective, but you have a lower cost and a lower hospital stay. My conclusion is, and the other conclusion is, half of the patients just need a drain to recover. My conclusion is you need both options, the minimally invasive surgical and the endoscopic, before you have a center that should treat these patients. For a simple reason, I give you an example. This is one of my patients. We put a lumen-opposing metal stand in here between the stomach and the necrosis with perfect drainage, but the drainage is not here in the retroperitoneum. It goes down in the entire retroperitoneum until the pelvis. And if you walk a pediatric endoscope through here for necrosectomy, and you go all the way down and you see the iliac artery in front of you, you should be a surgeon to be there, because this is not something, a complication, if you cause one, you can handle as an endoscopist. So these patients need additional drainage from the retroperitoneum, and sometimes they also need necrosectomy in addition to endoscopic drainage. So my recommendation is get your surgeons involved in this if you are a gastroenterologist, and if you're a surgeon, get your gastroenterologist involved in this. You need an entire team. Second half of my talk is more concerned with why do we develop necrosis and why do Astana cells die? And the hypothesis is quite old and from not far from here. Hans Chiari, an Austrian a pathologist at the German University in Prague, he proposed over 100 years ago that autodigestion of the pancreas by its own digestive proteases, trypsin was already known at that time, is the cause of pancreatitis. We've taken up this idea a while ago, and we developed a technique in isolated acini, usually of mice and rats, but sometimes of human, and we could detect this premature activation of digestive enzymes. This here is trypsin, this is elastase, inside living cells. So this is not a cut section with immunofluorescence. These are substrates. So they show us actual activity, and much of the data I'm going to show you are based on this technique that was developed in our lab. The opposing hypothesis is this has nothing to do with protease activation. It has something to do with inflammatory cell activation, leukocyte activation, and that one was proposed by Heini Rinderknecht, a Swiss biochemist working at the University of Southern California. Why do we think that this inflammatory process is so important? Because if you measure cytokines, here an anti-inflammatory cytokine, interleukin 10, or a pro-inflammatory cytokine, interleukin 6, they are very high at the beginning of pancreatitis. This is at admission. We don't know what they were before. However, they correlate very well with the severity of the disease. The higher the cytokine storm, SIRS, we call it septic inflammatory response syndrome, the more the severe pancreatitis is, the more likely the patient is to have necrosis, and the more likely he is to develop infected necrosis. So that plays a part. So what cells do make these cytokines and are responsible for necrosis? This is the family tree of immune cells. They start with the stem cell, and I'm going to point out three of them. One is neutrophil granulocytes, and I will talk a very little bit about their actual role in pancreatitis. The other one is macrophages, and this is what my group has studied over the last few years, and I think they have a very interesting role in pancreatitis. And the last one is T cells, and this is premature work, and we don't have enough time, so I will cut out on the T cells. What about the neutrophils? We and others, we were not the first to do this. Depleted neutrophils, you can do that by giving anti-neutrophil serum. So you deplete them from the blood circulation, and what you find in experimental models, this is a severe model of pancreatitis, if you take out the neutrophils, then you greatly reduce the activation of trypsin, intracellular activation. This was the first time we could show that the intracellular activation of digestive enzymes is not only triggered by chemical stimuli, but by inflammatory cells directly, and you also reduce the severity of pancreatitis. So it's not a question who is first. Probably you have two ways of activating your digestive enzymes. One is directly in the Astana cells, from the Astana cells by external signals, and the other is through inflammatory cells, particularly here at 8 hours. That's important. What's the role of neutrophils? And here I show you that neutrophils have usually, it's innate immunity against bacteria. You don't need it to be primed. It doesn't have to be learned. It's the same in the mouse and man. Neutrophils make use of histone citronylation, that's the protein that packs DNA, via peptidyl arginine deaminase when in contact with particular agents such as bacteria, and they extrude their condensed chromatin, that's the protein that binds DNA, and they form fishing nets to catch bacteria. It's called extracellular traps. This has been discovered 10 years ago, but what does it have to do with pancreatitis? If you look for these nets and neutrophils in pancreatitis, This is our duct ligation model plus stimulation of pancreatitis. You find neutrophils inside ducts, here again. In an experimental model, which I will allude to later, with IL-17 overexpression, you again find them into the ducts. You also find it in the human duct, and you find it in autoimmune pancreatitis. If you're familiar with autoimmune pancreatitis, it's a very interesting disease. One is IgG4 related, most prominent in Japan, and the European variety is called AIP2, autoimmune pancreatitis 2, and it's characterized by granulocytic epithelial lesions. These are these little balls inside the pancreatic duct. This is what the pathologist describes. It's actually neutrophil extracellular traps. A group in Erlangen, Moritz Lepkes from Erlangen, he's in the group of Marcus Neurath, they made use of that. They found these granulocytes with IL-17 inside the human pancreas. We sent them chronic pancreatitis tissue, and they find it in human material. Then they said, if it's IL-17 driven, usually you have naive T cells. They're stimulated by interleukin-23. They stimulate Th17 helper cells, and then IL-17 stimulates the neutrophils. They took a shortcut. They made the gut and other organs produce massive amounts of Th17, so you had a constant stimulation of neutrophils inside these animals. And they developed spontaneous pancreatitis. And if you look at that, oops, my cursor is not where I want it to be. So you find in this model, in this spontaneous pancreatitis, you find IL-17 positive cells. Then they looked in this pancreatitis model, and they could show that these granulocytes, they form plaques inside the little ducts of the pancreas. These plaques contain free DNA, not intracellular, free DNA. It's citrullinated, which is a sign for these extracellular traps. It co-localizes with epithelial cell markers and with PNM elastase, so they know it's neutrophil. That was proof that neutrophils go into the duct and they form extracellular traps. And what this does is it occludes the little ducts. So if you characterize it, the role of neutrophils, cells, to summarize their role, cells containing aggregates accumulate within the pancreatic ductal system under experimental conditions in chronic pancreatitis and in autoimmune pancreatitis. And the partial obstruction by these aggregates, which form extracellular traps, could be the trigger for pancreatitis or pancreatic damage. That was the story on neutrophils. What about macrophages? If you look at human pancreatitis tissue, or at this experimental model we use, you find this is stained with two different markers. The red marker is CD68, it's a macrophage marker. And the green is trypsin. So these are normal Astana cells, tiny here, and these are macrophages. And then Matthias Sandler, now a group leader in my department, and at that time a PhD student, he looked at those and says, there is area of colocalization. There is trypsin inside macrophages. I wonder what that does. I said, well, they're just cleaning up stuff. And did a number of in vitro experiments. So he took macrophages isolated from bone marrow and added SNI isolated from the pancreas together. And he made a very interesting observation. These macrophages, when they eat the debris or the components of Astana cells, they activate trypsin. It's not trypsin activation in the Astana cells, it's trypsin activation in the macrophages where it doesn't belong. And they do this in a way that is inhibitable with the trypsin inhibitor, but also inhibitable with the cathepsin B. This is a lysosomal enzyme that can activate trypsin in the pancreas. And the question, my next question was, do they only do this when they eat trypsin, or when they eat Astana cells, or also when they eat zymogen granules? And he added zymogen granules to macrophages, And the interesting thing, if you do that, this is the addition of acinar cells, this is the addition of zymogen granules, and both of them actually activate macrophages in a manner that is comparable to that of LPS. LPS is lipopolysaccharide. This is the strongest stimulator of macrophages, usually coming from bacteria. So acinar cells, when in contact with macrophages, are massively activated, and this depends on trypsin activation inside these macrophages. So to make a long story short, we went back to the animal model. This is the model we used. You partially ligate the pancreatic duct, and then you stimulate the animals with a secretagogue. And 2 days after you induce pancreatitis in this manner, he transferred macrophages into these animals. And he did so in cathepsin B wild-type animals and in cathepsin B knockout animals. And this is the result of this. If you take knockout animals and you transfer knockout macrophages, nothing much happens. If you transfer wild-type macrophages, you get massive activation, and this is dependent on and this regulates the severity, and this depends on the cathepsin B and the trypsin in these cells. This is my concluding slide. What do we think happens with the macrophages? Acinar cells lose zymogen granules or they die by an initial stimulus. This is phagocytosed into the acinar cells. I haven't shown you the experiments, but we show this as endocytosis. It's activated, trypsin is activated in lysosomes. It activates NF-kappa B inside the nucleus, and this is what drives TNF-alpha and IL-6 stimulation, which again leads to additional damage, and this is the circle of death for an acinar cells. So fighting pancreatitis probably doesn't mean prevention of trypsin activation inside acinar cells, but controlling inflammation. These are the people I collaborated with for many years, and the data I've shown you are mostly generated by Matthias Sendler, who did the entire macrophage story, by Ali Akdassi, who worked on the cathepsin, by Julia Meile, who initiated this program, Uli Weiss, our geneticist, and Moritz Lepke. You can tell which one is the MD and which one is the PhD. This is the PhD with the Sons of Anarchy t-shirt, and this is the MD with the tie. Moritz works in Markus Neurath's lab, and I thank you very much for your attention. If you're interested in the pancreas, in June we have the 50th EPC meeting in Berlin, and if you go to the website, the program is already on the website. I think it's going to be a very interesting meeting. Thank you for having me in Hermannstadt.